Thanks a lot for introduction. Thanks, Michael, for giving opportunity, like many times being attendance, sitting in the crowd silently. But now I think we have good chance to present some at least nice structure. Unfortunately, we are not about in polymers in this project yet, or we are there, but maybe give us some time. So today we'll talk rather about classics nanodisk system. So nowadays we are using for many years, but mostly for protein transport systems. But today it's kind of one of premier talks of our new development in polysaccharide transport. So wait a sec, why my slides? Yeah, here it is. So actually, it was one big entry for me, for my group, to change the focus, moving from protein secretion to something new and totally underexplored. So we talk about polysaccharide transport, which is required for biofilm assembly for bacterial communities growth. And biofilms say there are a number of publications claiming that it's a most abundant life form on Earth because like 50 to 80% of microorganisms, they actually live in biofilms, most cases invisible or visible. If you look in this picture, for example, in this pipe, that there is quite a microbial network community, not necessarily single type of microorganisms, but they efficiently live together. And what keeps them together is this massive amount of matrix, which is built of extracellular polymers. And here, the polysaccharides is the most predominant component. So there we also have some extracellular DNA, which comes from the cell, which was lysed, number of structural proteins present. So most cases those have amyloid shape and that they provide mechanical strength. And why interesting? So biofilms so highly abundant and not surprising that they have big connection to pathogenicity because it provides safe environment for the cells to multiply. They allow to exchange the DNA to develop resistance and also seen as extracellular proteolytic system. So basically a lot of nutrients can be captured by those polymers also and then be offered to bacteria. Um, it's important to know how to combat, or it would be really great to know how to combat biofilm beyond just simple mechanical stretching. So if you could block biofilm formation, for example, by interfering with secretion systems, that would potentially have medical benefits. And there we would need to know the mechanisms. And it's notable that so far there is only one well-studied secretion system for polysaccharide. That's a bacterial cellulose, extensive work done by Petya Krasteva in Bordeaux and Johann Zimmer in the US. The structure is known, the mechanism is pretty well understood, but that's so far that's the only system which have been clarified. But if you look at the protein secretion machinery, we have now 10 secretion complexes, secretion systems, pretty well understood. So clearly polysaccharide transport has a lot still discoveries ahead. And that's where we targeted our research last few years. So we look at the polysaccharide called PEL, originating from this infamous pathogen Pseudomonas aeruginosa. PEL stands for pellicle, so those tiny films which are formed at the liquid surface by bacteria, but also PEL is required for biofilm assembly on surfaces. So it's not the polymer which is pathogenic in the human when for example, bacteria multiplies in lungs, but that's polymer which allows cell to occupy new spaces in the environment. What is known about PEL that it's a linear polymer, partially charged, so partially cationic, so it has positive charge, and then it can interact electrostatically with extracellular DNA and contributing to matrix production. What also known, we have idea about all the components which are involved in synthesis and secretion. So that's drawing of machinery. So inner membrane complex would have synthase, would have some regulatory subunits, and surely it would have a transporter in the membrane. And then polysaccharide goes through the periplasm, it gets partially functionalized, and then it should be exported via this barrel, presumably barrel in the outer membrane. Our problem is that this drawing summarizes most of things which we know. That basically we know how things work, we potentially know how they interact, but structure is really missing. So it's very interesting system with a lot of components, regulation, modifications, but knowledge is rather limited. And it's not, though the polysaccharide is not found only in Pseudomonas, but also in a number of other organisms, including thermophiles, for example, those which live at 70 degrees or even 130 degrees. Those organisms, they have extremely small genome, but this the whole operon is present there, suggesting that this polysaccharide is very important for cell viability and survival in those conditions. So learning something about it would be really interesting. And that's a hero of the day, Marius Benedens, now a PhD student in our lab. And the results presented here, that was actually his master project, which developed extremely fast from cloning to cryo-EM structure. And I have to say that this project was a desperation project because we spent several years trying to analyze the inner membrane complex, this beautiful, colorful machinery, with rather not really fascinating results. And then when we needed to think about some new direction, so new student is joining, we consider that maybe we can look at the outer membrane barrier, this outer membrane exporter. And I have to say that we never touched outer membrane protein before in history of our lab. But cool thing about this membrane exporter, so-called PelB, the blue subunit, but B stands for not for blue, but that's actually, it's not only in outer membrane protein. So if you look at the structure, it has a predicted barrel, then it has very long scaffold from alpha helices, which is long enough to protrude the whole periplasm. So it's potentially a physical bridge used by polymer to travel. And this red domain in the N-terminal, it has all the properties of classical transmembrane helix for inner membrane. It has no cleavage site. So it looks like it's a rare example where when the protein would be anchored in both membranes, and potentially it interacts with inner membrane complex and it guides the polysaccharide directly for export. And it's not only the barrel which you find in the membrane, also there is this lipoprotein PelC indicated in orange, and that's element where we know the structure from crystallography previously, group of Lynn Howell in Canada. It shows a ring of 12 symmetrical subunit crystal structure. Everything is pretty much symmetrical. And it's known that this ring is essential. We don't know why. And we even don't know if this 12 subunit symmetry remains for the whole complex, because if we feed it into AlphaFold, it generates a structure, it generates a model for 12:1 ratio, but also it generates a model to 11:1 ratio, and it generates nice model of 13:1 ratio. And you see that in some cases, maybe some contacts are missing, but also it stands for 12. So maybe 11 is actually would be a case, and that's has been shown for other outer membrane proteins, for lipoproteins, that stoichiometry in crystal phase, in crystallography and actual may differ. So we consider that let's hunt for this totally unknown complex. So we established very elegant, I really like the system that we, again, first time for us to look at the outer membrane. We put two genes in the same vector and consider that every protein PelC and PelB, they have different roads how they reach outer membrane. So one is lipoprotein, so it has this LOL pathway, and PelB is probably targeted via BAM. So we encode them both with specific signal sequences in a vector with purification tag, and it was quite encouraging from beginning to see that if we separate inner and outer membranes using the sucrose gradient, then we see PelB and PelC in high sucrose fraction where outer membranes are expected. So they nicely co-migrate with OMP-A, a marker in Escherichia coli. We could also solubilize a protein, the whole complex, seemingly with classical detergent like DDM, and use the histidine tag on PelB to pull on it. And then we see that in elution fraction, both PelB and PelC bands are visible. And when injecting it on size exclusion chromatogram, we see that indeed some big complex has been assembled. not only in peak intensity, but also if you look at elution volume, we talk about hundreds of kilodalton. And on the SDS page, we see again bands of two components. Remarkably, we look at the later fractions, we see PelC alone. And what it tells us that actually PelC should not be purified initially, but it tells us that complex is unstable in detergent. It falls apart, and indeed, longer we keep it, less complex we could isolate. So then we rapidly reconstituted it in two systems, nanodiscs and amphipoles, and submitted it for cryo-electron microscopy to Munich, to a group of Roland Beckman with extremely talented PhD student Christian Hernandez. So they look first on amphipole. His was sample shot day before Christmas, and so on the 1st or 2nd of January we got the first reconstruction. So you see immediately in 2D classes that the ring appearing, and one can even on these raw images, raw classification count that indeed 12 subunits are present there. And then reconstruction was delivered showing indeed ring is there, but the barrel is not sufficiently resolved. And if you look carefully at these 2D classifications, it explains why. We have nearly exclusively top and bottom views, side projections are missing. And that's why we do not get idea how the barrel looks like. So it was a big drama, trials to maybe collect more data, but at the same time, nanodisc sample was shot. And here it's completely different story. We have rather uniform projection from all directions, which again was without much controls done before. It was shot over the weekend and resulting in a week in 2.5 angstrom structure where we can see the ring, we can nicely see the barrel, and we see the lipid density around. So that's the overview showing off with the resolution that we can see not only aromatic residues, but also rather small ones. We see all the structural element, and then we got the structure. We have indeed, as expected, a barrel. Then we have a ring of 12 subunits surrounding it. We see the interaction that this helical scaffold, it has some interactions with PelC, with the ring. And then we start comparing it with the structure. So what we actually learn already from this. So in contrast to cryo-EM, we visualize all the parts of the protein, including the helices, which lay on top, and then they clearly change the electrostatic of the protein. So crystallography suggested that this heavily red area, so negatively charged area, it will be exposed to polysaccharide and drive the charged polysaccharide into the barrel. So cryo-EM tells like, no, actually this highly negative charge will be already covered by a loop from the top. Maybe there will be some charge accumulated in the middle, but probably it's not so important. So previous functional studies from Lynn Howell lab suggested that the charge is important. So they generated number of mutants and showed that particular aspartate in this PelC ring, once the charge is inverted, the biofilm formation is nearly abolished, which tells that either polysaccharide transport is deficient or maybe the PelBC complex is not assembled. So we look at this residue or region where aspartate 119 is located. So that is the loop of PelC subunit, and you remember that crystal structure reported a uniform symmetric assembly. For us, we see that each individual subunit has its own organization, so this loop is always bent a little bit. So why would it be so? You consider where this loop is exposed. It's not facing the polysaccharide, but it's actually facing the barrel. So this negative charge shown here, it's not involved in polysaccharide transport, but it's involved in forming the complex. So nearly every aspartate finds a partner at the rim of the beta barrel. Either it will be arginine, maybe it will be some part of the backbone, but there are a number of electrostatic interactions formed here which keep the complex together. So if you look deeper, that it's not only electrostatic interaction what's important, but it's also number of lipids we can visualize. Those green densities, they do not belong to the protein, but those are easily identified acyl chains. So for example, those are acyl chains of lipoproteins. So we can see all the three lipid anchors, and they're always for each subunit, they're sort of clamped by two tryptophans on the sides. So it provides apparently extra stability for the complex. So we have electrostatics, we have the acyl chains which hold it in the membrane, and we have those tryptophans which keep the lipids well packed. We see more lipids. We see, for example, those very long densities which go in between of rows of aromatic residues on beta barrel. So actually looking at the beta barrel for the first time in life, it's like shows really amazing architecture. So it's so nicely fine-tuned for protein lipid interactions. And also there are those spots of charge where we think it, so that's extracellular side of the protein. That's where the lipid A will potentially bind. So we look at classical phospholipids in our nanodisk, and then so we provide those, so we visualize phospholipids at the end. But I guess with polymers, there will be a chance to look at them. LPS. Yes, Michael, I see your sign. So then just briefly, so we look at we have idea how it's assembled, but it still doesn't tell us how it functions. So in the structure, we see several loops which are closing the barrel and would not let polysaccharide to go through. So some dynamics would be needed. So we look carefully at individual loops which close it, and then we suggest it was clear from the structure that maybe plug S, rather short loop, which is contacting the essential tyrosine, which may screen the polysaccharide chain. So that probably will be the loop which can be displaced. So then we can skip this part. And then we were lucky to recruit Jennifer Loschwitz to do a number of MD simulations for us. So because we can do cryo-EM and rather classical membranes, but what about different salts? What about LPS present? So that's what we can analyze with MD simulation and see maybe the protein dynamic is affected. Maybe we capture opening event. It shows that membrane actually doesn't do much. It doesn't have some specific lipids have not much effect, but indeed transient opening of rather broad pore can be observed in the particular moments of simulations, and those opening will be indeed enough to translocate the whole polysaccharide through, and that those openings are primarily related to plug S dynamics. In the final proof of this dynamic, we were lucky that University of Groningen group of Giovanni Maglia helped us to look at transient opening of the pore with electrophysiology experiments. So they look at individual PelB molecules in synthetic membrane and see if current goes through or not. Briefly saying, the data was hard because it's single molecule measurement. Apparently, each molecule demonstrates its own behavior. And then to clarify actually which loop is most important, we simply deleted those. And once we touch the plug S, we see clearly changing behaviors that the channel was permanently open. The noise was gone, so meaning that this plug S, the short loop, does not fluctuate anymore. And indeed, as model prediction and modeling shows that there will be a channel formed. So at the end, from this pretty much one data set from cryo-EM and then follow-up MD and single channel recordings, we get idea about the structure, we get idea how the whole complex get together, and we have preliminary model. Now data were supported for the polysaccharide transport, that it will enter into the negatively charged barrel, the plug S will open, and then the polysaccharide will stream. We still have no idea why C ring is required. It's not conserved evolutionary. The barrel is C ring net. No, maybe it supports to dock enzyme A. We don't know exactly interaction with native membrane, so that's where we're betting now on SMALP and ultra amphipoles, and surely PelBC interaction with substrate would be next frontier. And then acknowledging people, Marius, Jennifer, all the collaborators, funding agencies, you for your attention and patience. Thank you much.
Alexej, thank you so much for your talk. I love the visuals of your presentation, and we really look forward to learning more about the native lipids once you move over to polymers.
So I will I will give the spoiler. Yes, we extracted it nicely with one of those ultra poles and looked nice and negative stain. Somehow we do not see a single particle in cryo-EM. We don't know what goes wrong. After 2 days collection, there was nothing. Maybe Philip can comment on this anecdotal experience.
Luckily, the native MP screening kit allowed you to try out all the polymers. So maybe that's the best next step. Since we are running a little bit, we're on the edge of time. There's one question. After that, if there are any other questions for Alexej, please feel free to pop them in the chat, and then you can just answer from there. I'll also just quickly remind all speakers that the timing is 15 minutes for a talk, and then we have 5 minutes for questions, so moving forward. But other than that, Alexej, the question from Michael would be, Michael, would you like to unmute yourself and ask your own question?
Sure. Great talk. Just wondering if you saw any contacts between the PEL proteins and the MSP that might not just stabilize the barrel and the lipids, but also maybe affect the functional integrity of the protein.
I had impressions that you were a reviewer of our manuscript. No, we haven't seen. So basically we have all this range of MSPs. So here it was MSP 1D1 used because that was sufficiently big not only for the barrel but also for PelC. We later reconstituted also an MSP 2 and 2 for completely different purposes. We see totally identical structure. Even the lipid density, this long lipid or this long rod appears, because I was really afraid that it's artifact. And first I was also thinking for 2 days, what lipid can actually span the whole membrane? I just realized that it's just overlay of 2 acyl chains. But no, so there is no influence of lipid of disk dimensions here. Thanks.